Hey Beaks, it's Mr. Boylan, and today I want to talk to you about mutations. So it's really important that you understand gene expression before you watch this. So if you have to go back and watch the video on transcription and translation, maybe you should do that first. But if you feel good about gene expression, this should make sense to you. So let's go. So mutations basically can happen randomly. Okay, it's just one of those things, and a lot of times they don't happen within a gene, so we'll never even know about them. But there are some agents out there that cause mutations. These are called mutagens. Right, physical or chemical agents that can cause mutations. The ones that we talk about the most are carcinogens, right? Those are very important, and those mutagens can cause cancer. So mutations of one or a few nucleotides can affect protein structure and function. Remember, that's the whole central dogma of biology that we talked about. The DNA tells you how to make your RNA, and the RNA tells you how to make your proteins. So if your DNA is messed up, that's going to goof up your gene expression and goof up your proteins. So mutations are changes in the genetic material of a cell or a virus, right? Right now we're dealing with the coronavirus outbreak, right? Of not good, and we're worried that, okay, hopefully we'll get a vaccine soon, but if the virus mutates considerably, well then possibly our vaccine won't work anyway. But point mutations are chemical changes in just one base pair of a gene. The change of a single nucleotide and DNA template strand can lead to production of an abnormal protein. So this can just be one little tiny change, and that could cause some serious problems. Let's see here. So here you can see wild type hemoglobin. So all of us, this is a quaternary protein hemoglobin right here. It's made out of 4 separate polypeptide chains coming together, and the beta chain of this, normally what we have inside of our DNA, inside of one of the genes here, is CTTGAA. Okay, so this is the template strand up here, and the mRNA that'll be transcribed is GAA, and if you use your decoder, we know that that's going to be, okay, glutamic acid. So, people that have the sickle cell trait or carry the sickle cell trait, their DNA, instead of saying CTT, says CAT, right? So, that is going to create a different transcript here, or a different piece of mRNA, and now the reading frame, instead of saying GAA, says GUA, and that's going to be valine. Okay, so what's the major issue there? Excuse me. Don't forget, proteins, the tertiary structure, in this case quaternary structure of a protein, the shape dictates the function. So it's really important to have that ultra-specific 3D shape correct. And in this case, because of the different R group, right, glutamic acid and valine have very different R groups, and because the valine got substituted in, the protein changes shape, and that can cause the disease, which we know, sickle cell. So, point mutations within a gene can be divided into 2 general categories. You have a nucleotide pair substitution, or one or more insertions or deletions. Okay, and these are different things, so we have to wrap our head around it. Generally, you know what this word means. Substitution just means you swapped one letter for another. Okay, and they can be bad, like we just saw in sickle cell, or you might get away with this one pretty well. Now these, insertion or deletion, another way to call them, are reading frame mutations, because they're going to shift the codon reading frame sequence. Okay, so those are a little tougher to deal with generally. So here we go. A nucleotide pair substitution replaces one nucleotide and its partner with another pair of nucleotides. Okay, so if we're looking up here, we can see some examples of wild type, okay, is given to us, and this is normal. So we're just looking inside of a random gene here, and we have, based on the DNA, and then the RNA, use the transcript of DNA to make that, and then here's our protein, or our amino acid chain. All right, sure. So we have methionine, lysine, phenylalanine, glycine. Okay, so this is all normal, and then eventually we hit a stop. Okay, a stop codon right here, and that's going to stop it. So, what if we had something called a silent mutation? Silent mutation had no effect on the amino acid produced by the codon because of the redundancy in the code. Okay, do you remember when we were talking in our other video about the first 2 bases of the code? Here comes the tRNA, the anticodon, and the first 2 have to lock in pretty well. But the third, that wobble pair, that doesn't matter quite as much. That's kind of what's going on here. You can see that in a silent mutation, we do have a substitution. Okay, so right here we should have CCG, but now we have CCA. Okay, so we have an A instead of a G. So what's happening is that mutation right there, well, what's it gonna do to our mRNA? Well, as you can see now, our mRNA, instead of saying, excuse me, GGC, is going to say GGU. Okay, but our amino acid, glycine, is still glycine. Oh, so in other words, if we used our decoder, GGC and GGU are the exact same amino acid. So that didn't matter whatsoever, and that's why we call it silent. There's no effect on the amino acid sequence. All right, well, that's good. Let's look at the next one here. Missense. Okay, missense mutations still code for an amino acid, but not the correct amino acid. All right, so let's take a look at this one. We know that up here again in the wild type we have AAA CCG. Well, look what happened here. AAA TCG. Okay, so here we have a T instead of a C, and this one's a bit different because this change is at the first nucleotide of the codon, right? So when the tRNA comes down, no, you're not going to have the right tRNA anymore. You're going to have a different tRNA to bond to that, right? So if you think about it here, now we have ACG for my mRNA, and that's going to code for a completely different amino acid. In this case, we were supposed to have glycine here, and now all of a sudden we have serine. Okay, now you might think to yourself, oh, that's just one little change, right? Does that really make a huge difference? Maybe not, right? And there's a couple reasons maybe not. Maybe this amino acid inside the grand scheme of the whole protein isn't at a spot that's really important for the shape of it. Or maybe it's not in like the active site of the enzyme or something. Yeah, you might get away with it. Or it could be like sickle cell where, no, this could kill you, right? So it depends where it's at. Another thing is, what if that R group that changed, right? So what if we change one amino acid for another amino acid, but the R groups between them were so similar? Sometimes you get away with it there too, right? So missense mutations, they can be bad, right? Because you actually did code for the incorrect amino acid. Okay. Now, nonsense mutations, these are generally a little scary. What's going to happen here is a change in the amino acid codon into a stop codon. Okay, so this is probably going to lead to a protein that doesn't function. So, for example, as we're moving along here, you know that we have TAC, TTC, TAC, ATC. All right, so in this case, here is our A instead of a T. And well, what's going to happen now is we have UAG, and UAG is one of those three stop codons. So here we have, I don't know if this would be considered the start codon in this case, but because it'd be very short, but regardless, methionine, and now stop. Oh boy, that's nonsense, right? So a nonsense mutation, substitution mutation, that simply means that we're moving along and, oh, because we substituted one letter for another letter in our nucleotides, we hit an early stop codon. Not good. Okay. Okay. So now we move on to the more drastic changes, right? We just spent some time talking about if you substitute one letter for another letter, and that could be pretty tame results, or it could change a lot, right? Well, this one is going to change a lot, because what we're going to do here is we're going to put another letter into the DNA or take one out of the DNA. And what that's going to do, if you think about it, if it's in the coding sequence or inside the exons for eukaryotes, it's going to shift everything, right? So here was my normal 1, 2, 3, right? Here was a codon. If I take one in or out, you know, all of a sudden that's going to shift. And now everything else will shift as well. So these can be pretty dangerous. So insertion or deletion or reading frame mutations. So insertions or deletions are additions or losses of nucleotide pairs in a gene. These mutations have a disastrous effect on the resulting protein more often than substitutions do. So let's see here. Here's an insertion or a deletion. So here we have TAC, and we are supposed to have TTC, right? Well, TTC is over here. TAC is, oh, we added adenine right here. Okay. Well, because we added adenine, instead of us reading TAC TTC, we're reading TAC ATT, right? CAA. What is going on here? Well, let's take a look at the mRNA. AUG, UAA, AUG is start, UAA, as you know, is a stop codon. Now, what's the deal with this stop codon? Well, if you remember, that's nonsense. When you hit an early stop codon, that's nonsense. So we actually inserted a letter and immediately hit a stop codon because of it, because the entire reading frame got shifted. Okay, not good. Okay, and now if you look down here, frame shift, what's going on? We are missing an A, right? We used to have, up in a wild type here, AAA, CCG. Now we have AA, okay, no more A, C, CGA. So now we have to read it AAC, CGA, which means when we make our mRNA, instead of UUU, which would have been phenylalanine up here, now we have UUG, which is leucine. And now instead of GGC for glycine, we have GCU, which is alanine. Do you see? Because we are missing a nucleotide from our DNA, we're now going to be missing that nucleotide in our RNA, and that's going to shift the entire reading frame over. Okay, and now every single amino acid that we have after that is going to be different. Okay, so these frame shift mutations, not necessarily a good thing, right? Now, just like before, we can actually get lucky here. Okay, and I don't know if lucky is the right word, but here you can see we normally have TAC, TTC, AAA. Well, look what happened down here. TAC, oh, TTC completely is missing, and now AAA. What that did was that removed an entire codon. Okay, so if an entire codon is removed, that's interesting. It's still a frame shift. Everything's still going to get shifted over, but because we removed 3 bases, you're still going to have methionine, phenylalanine, and glycine. Oh, so we're just missing a lysine right here. Normally we'd have methionine, lysine, phenylalanine, glycine. Now we have methionine right to phenylalanine. So this could be very bad, or maybe we're okay missing that one amino acid. Okay, but in the grand scheme of things, at least we didn't change the rest of the chain. Okay, so sometimes reading frame mutations, we can get lucky with them, but those are a little bit worse. So what did we learn about here today? Well, we learned that you have the wild type of your DNA, right? So this is what we all have, hopefully. But you could substitute a letter, and if you do substitute one of those letters, there's a couple things that can happen. The first thing is, well, nothing can happen, right? You can have the exact same amino acid, and that's because of the redundancy of the genetic code. Okay. The other thing that we talked about that could happen is, well, you could substitute a letter and then you could have a different amino acid. Okay, and that could be dangerous, obviously. And you could also substitute a letter and hit an early stop codon because of that. Okay, and that's nonsense. So silent, no effect, redundancy. Missense, new amino acid. Nonsense, you hit an early stop codon. Okay, but these, okay, over here, now instead of a substitution, we're talking about an insertion or a deletion, and that leads to a frame shift mutation, and those are a little bit more dangerous, right? Because in this case, we shifted the frame, and now we hit an early stop codon. That's no good. In this case, we took a letter out, and now all of our amino acids are going to be different, right? And down at the bottom here, we did remove an entire codon, and hey, that helps for the rest of the amino acids because at least they'll stay the same because we still have the same reading frame because we removed 3, right? But regardless, that's a little bit about mutations, and I hope you guys found that helpful. Okay, good luck. Take care.